So if you check out this video, essentially what it's showing is the pattern of reabsorption in the loop of Henle. So here's our filtrate, what we would lose if it stayed in the nephron. But if you notice, is as the filtrate comes down the descending limb of the loop of Henle, water is lost that is reabsorbed from the filtrate. So we're taking water that is in the filtrate and keeping it either in the interstitial space of the kidney or in the bloodstream around the loop of Henle as well. So we're getting it back into the body. And what we know about water is that it only goes towards places of higher osmolarity. And so the reason it's leaving the filtrate, is if you notice, is that this interstitial space around here is at an extremely higher and higher osmolarity as you go deeper and deeper into the kidney. So as water is lost, the filtrate becomes more concentrated, but water keeps getting lost because this tissue out here is even more concentrated. So we lose water, this gets more yellow, but we keep losing water because the concentration of solute out here is so high. So what these numbers are showing is the osmolarity in the tissue. So as the filtrate goes deeper and deeper, the osmolarity just gets more and more, and that keeps pulling out water as it goes towards higher osmolarity. So essentially, water is reabsorbed from the filtrate here due to the high osmolarity in the interstitial space of the kidney. All right, so what is setting up this osmolarity gradient then? If this is what's driving water reabsorption, what is driving this? What is setting up this gradient? And that's where the ascending limb comes into play. If you notice here, the ascending limb is the kind of the opposite of the descending limb. It does not allow water to leave or to enter the filtrate, but what it does do is pump out sodium chloride. So if you notice, as the filtrate comes up the ascending limb, sodium and chloride are pumped out into the interstitial space. This pumping of sodium chloride out is what allows this concentration gradient to stay high on the outside of the filtrate, on the outside of the nephron. So essentially, what is happening here on this end, on the ascending limb, is setting up what is happening on this side. Because the ascending limb sets up this concentration gradient, all this salt on the outside of the nephron, that's what drives reabsorption of all this water on the descending limb. So this causes this. So this solute concentration gradient in the interstitial space is critical to allow water to be reabsorbed, which helps concentrate the urine and prevent all this water loss. But if we kind of think about it, what was happening is that we were losing water to the interstitial space in the descending limb. So why doesn't all this water that's lost from the filtrate dilute the solute gradient that we set up? In other words, what we're saying here is, okay, we lost salt out here, and that allowed water to be reabsorbed. But why doesn't the water that got reabsorbed here just completely abolish this gradient? Because if water is leaving, wouldn't it just dilute this osmolarity, make it less concentrated, and then no more water would be reabsorbed? Does that make sense? So essentially what we need to do is to somehow prevent this water that's being lost from messing up this gradient. So why doesn't the descending limb loss of water basically destroy the solute gradient in the interstitial space of the kidney? And here's why. The answer is that that water that's being lost from this descending limb is being removed by this blood vessel, this network of capillaries around the nephron called the vasa recta. That is, the water that leaves from here to here then goes into the blood so that it doesn't stay here and dilute the concentration gradient. So any water that's lost is going to be swept away into the bloodstream. So that's one reason why the water does not dilute the concentration gradient, because it goes into the blood and is removed from the interstitial space. Another reason, if you notice something weird here, if you follow these arrows, you'll notice that the filtrate is flowing in this direction. So it's going down here and then up here. If you look at the blood flow though, it's going in the opposite direction. So as filtrate is flowing up, the blood is flowing down on that side, on the ascending limb. Here, as the filtrate is going down, the blood is actually flowing back up. And whenever you see something funky like this in physio, there's always a reason. Your body is designed in a certain way for a very important reason. And so what we're going to ask is, why is there this what's called counter current, where the filtrate and the blood are actually flowing in opposite directions? It's a super, super important reason. And here's why. Essentially what we're going to do, so this is showing this exact same picture, showing the nephron, the loop of Henle, and then the blood vessel flowing in the opposite direction. And as a reminder, what we want to do is to take water that was lost from the descending limb, get it into the bloodstream. But remember that water only flows towards regions of higher osmolarity. So to get water from here to here, that must mean that the osmolarity at this point in the blood is high, because water needs to go there, will only go there if the osmolarity is higher. Make sure you're convinced of that. So basically anywhere water is going, that must mean that's going from low to high osmolarity. And here it's going from low to higher osmolarity. And the question is, why does the osmolarity here in the blood, why is it so high? And the reason is because of this counter current system. So if we follow the blood is coming down, and what it's going to do is absorb some of this sodium and chloride and also potassium that was pumped out by the ascending limb. But if it absorbs some of that sodium chloride, as it absorbs more and more as it's going down, that means that the osmolarity of the blood when it gets down here is now super high, because it's taken all this sodium chloride and absorbed it into the blood. And the reason that is so important is because once the osmolarity of this blood is really high, when the blood comes up this way, then it's going to be able to reabsorb the water. So essentially the blood over here gets really concentrated with salt, so that on this side it can reabsorb all that water. That process would not work if the blood and the filtrate worked in the same way, or if they flowed in the same way, in the same direction, I should say. So again, the idea, if we look at this image right here, is that as relatively low solute concentration blood comes in down next to the ascending limb, it picks up salt that was lost by the ascending limb, and as a result, the osmolarity of the blood gets higher, gets really high when it dips down at the bottom, and as it comes back up, that allows it to reabsorb the water lost by the descending limb. So this countercurrent exchange is necessary because it's basically setting up this reabsorption of water. The blood comes down in this direction, reabsorbing the salt that was lost by the ascending limb. When it does that, that allows for reabsorption on this side of the water from the descending limb. So again, it's kind of this sets up this. You good with that? I know you can't ask questions right now, but bring your questions on Monday. We can talk more about it. But again, the idea is that the fact that the filtrate and the blood are flowing in opposite directions is critical. Because what we can try thinking about is what would happen if the blood and the filtrate were flowing in the same direction. So for example, if the blood was coming down this way in the same direction that the filtrate was going. Let's see what would happen. So if the blood was relatively low osmolarity, low solute concentration, water was being lost from here into this interstitial space. As this blood comes down, there'd be no reason for water to go in here because the osmolarity of the blood would be low coming in. That means the water would stay right there, and the concentration gradient in here that we set up previously is now gone. And everything stops. Water reabsorption would then stop. So what we need to do is to make the blood really concentrated on this end and then bring it back up here to allow for the reabsorption of water. Alright, so now we've kind of discussed some of the details in the loop. The filtrate will then proceed through the distal convoluted tubule where some stuff will be reabsorbed, like sodium chloride, more water, bicarbonate. Some stuff will be secreted to be lost in the P, like potassium, H+. But what we're really going to do is focus more on how we regulate how much or how little water or how much or how little salt we reabsorb based on hormones. And as we talked about in the lab, the way we're going to regulate how much water or salt is reabsorbed is by regulating reabsorption at the later part of the distal convoluted tubule and in the collecting duct itself. So let's see how we do that. If you guys remember, we talked about these two hormones, ADH or anti-diuretic hormone. I kind of think of it as APH, anti-pee hormone. Basically, it's trying to prevent you from peeing out water. It's also known as vasopressin. And generally what it does, it helps increase H2O reabsorption. That is, it prevents diuresis or loss of water, or diuresis, just urine production. And we're going to see that it does that by increasing the expression of what are called aquaporins, or just channels or transporters for water. Literally called water or aqua pores, or holes, water holes, basically. This hormone is secreted from the posterior pituitary, as we know. And remember, it's not made in the posterior pituitary, it's actually made in the hypothalamus, just as some endocrine review. And we'll see later when you want to release it. We'll talk about that later. So basically, ADH affects water reabsorption. Aldosterone, on the other hand, its main goal is to affect sodium reabsorption. And we're going to see how that does that in a second. So let's look at ADH, or anti-pee hormone. If we look at how it acts, if you notice here, we get some clues about this hormone based on this mechanism. So what you notice is that this hormone is traveling free in the blood, does not need a protein carrier. That means that it is hydrophilic. It is perfectly happy in the watery substance of the plasma in the blood. But what that also means is that it hates fats. And as a result, it's going to need a cell surface receptor, in this case a GPCR, in order to signal to these cells to help reabsorb more water. And essentially what's happening is that this GPCR, like we know, is going to result in the production of a second messenger like cyclic AMP. And cyclic AMP is going to trigger these little vesicles that have these water channels on them to move to the surface of these collecting duct cells. And when they do, that means that now water has a door through which it can go from the filtrate into these cells, and from the cells be reabsorbed into the blood. So the way that ADH, anti-P hormone, or vasopressin affects water reabsorption is basically to increase the expression of water channels on the surface of these collecting duct cells. So in a way, it's a lot like insulin, where, if you guys remember, insulin was operating through a tyrosine kinase, so some differences, but basically insulin's job was to increase the expression of the glucose transporter to allow glucose to get into the cells. That's basically what ADH is doing right here. And so just keep in mind that if water is going from here to here, from here to here, from here to here, it's always following concentration gradient, the osmolarity gradient. And you can see that it's greatest in the blood, so water is going to go to there. If we look at aldosterone, what you notice is that aldosterone is floating in the bloodstream, and they're not showing it here, but it should be bound to a protein carrier. And the reason you know it should be bound to a protein carrier is if you notice it's able to permeate this cell membrane easily. That means that it is lipophilic, which means that it is extremely hydrophobic. And so it does not like being in the watery substance of the plasma. So just some endocrine review. But anyway, so let's say you release aldosterone. Aldosterone comes into these cells in the distal convoluted tubule. They're called P cells, but you don't need to worry about that. Aldosterone comes in, binds to an intracellular receptor, a receptor inside the cells. And what we said about these receptors is that what they tend to do is to increase transcription of certain genes to make new protein from scratch. That's exactly what aldosterone does in this case. But if we think about it, if aldosterone is supposed to increase sodium reabsorption from the filtrate, the proteins it's going to make are sodium channels. So aldosterone has this crazy effect where it's actually telling these cells to make new sodium channels from scratch, put them on the surface of this distal convoluted tubule cell to allow sodium to be reabsorbed. And you can see that aldosterone also causes potassium to be secreted, but we're not going to worry too much about that. So very similar. ADH increases doors or transporters for water. Aldosterone increases doors or transporters for sodium. And what you notice is that aldosterone, what it can also do is make new sodium-potassium pumps or increase the activity of ones that are already there to force this sodium to get reabsorbed against its gradient. Alright, so essentially both of these hormones are just increasing channels for specific molecules. ADH helps you pee less by opening doors for water to get out of the filtrate. Aldosterone helps reabsorb sodium by opening up more doors for sodium in the DCT. And so if we think about this dude, what we saw was that he was peeing out like crazy. But there was nothing wrong with his glucose levels. And what we just learned is that ADH is responsible for regulating how much or how little water is reabsorbed. in the collecting duct. So it turns out that Andy has a disease called diabetes insipidus. He does not produce any ADH, or his body does not respond to that ADH. And as a result, he has no water reabsorption in the later parts of the nephron, specifically the collecting ducts. And so he pees a bunch as a result of ADH deficiency. And if you notice, this disease also has this word diabetes. So just keep in mind that when somebody says diabetes, it does not mean glucose, elevated glucose levels necessarily. All diabetes means by itself is overproduction of urine. I think it's Greek or something for flow, like tons of flow. So in diabetes mellitus, so that's type 1 and type 2, we have crazy urine flow, but it's also high in glucose, so it's very sweet. That's why it's called mellitus. Mellitus comes from the word meaning sweet. Like in Spanish, you say like miel is I don't know, jelly or honey or some like sweet jam or something. So this is basically tons of flow that is super sweet. Diabetes insipidus, like Andy has, is tons of flow that has absolutely no flavor. So if you're ever curious, you can taste some pee and determine whether it's insipidus or mellitus, even if the volume of the urine is exactly the same. All right. So essentially what we have now is we review the anatomy of the nephron, so the different parts, the glomerulus, the Bowman's capsule, PCT, loop of Henle, DCT, collecting duct. We reviewed that different parts of the nephron can either reabsorb stuff from the filtrate or secrete it into the filtrate to either pee it out or prevent you from peeing it out. And then we learned that one way that we can affect how much urine output we have is through these two very powerful hormones, ADH and aldosterone. Okay, so now my favorite part of the whole class, probably, part 3. Regulating blood osmolarity, pressure, and volume using the kidneys. Here's why this is my favorite part. Because it is literally going to combine just about everything we've talked about the entire semester into one giant regulatory mechanism. And the reason you should kind of love this is because you already know like 75% of how this works. All we're going to do is put it together. So let's do that. So essentially what we're going to try to do with the kidneys is, one, regulate plasma osmolarity. We're going to make sure that our osmolarity is not too high and that our osmolarity is not too low, that it's just right. And if you guys remember, the reason osmolarity is so important is because if it gets out of whack, your red blood cells could explode. If it gets out of whack, cells in your tissues could either expand or shrink, depending on whether their surroundings are hypotonic, isotonic, hypertonic, whatever. So what we need to do is to make sure to maintain osmolarity to maintain proper cellular function. The kidneys are going to allow us to do that. So again, the reason this is so important is because if we F with osmolarity, we F with the proper function of these cells. So for example, if more potassium than normal starts leaving these cells, all of a sudden these cells become hyperpolarized, they become less likely to fire, they become less responsive. So properly maintaining the osmolarity allows these cells to maintain, for example, their membrane potential, their excitability, and their normal function. Another thing we're going to do with the kidneys is maintain fluid volume. Make sure that the blood volume throughout the cardiovascular system is sufficiently high. but we're also going to try to prevent it from being too high. So again, the definition of homeostasis is you're saying, I want to keep a certain value within a certain range. We want to make sure that blood volume is not too high, not too low. And the reason this is so important is because what we know is that blood volume directly affects blood pressure. If blood volume is too high, pressure on this blood vessel will be too high. That vessel could explode, and if it explodes in your brain, that's bad. And so we need it not to be too high. Similarly, if we need it not to be too low, because if pressure is too low, then we don't have sufficient pressure gradients to drive blood flow to vital organs. So these are two absolutely critical parameters that we need to regulate. Osmolarity to maintain the proper function of cells, and blood volume to maintain the pressure gradient to drive blood flow throughout the cardiovascular system. So what we're going to talk about is how your body senses changes in osmolarity, how it senses changes in blood volume, and then we're going to integrate those two together to see how your kidneys do both at the same time. So as an example, we're going to take a look at somebody who, say, has been hiking for 3 days in the desert. As a result, they've lost a lot of water from their body, which means they have a low blood volume, low blood pressure, and the blood that is remaining has a very high osmolarity. All right, so how can we basically fix this person's problem? Try to increase their blood volume and decrease their blood osmolarity. Again, you might not know it, but you actually kind of already know how to do this. So if we think about it, we need to fix two parameters via homeostatic feedback loop. And what we know for any good homeostatic feedback loop is we need a sensor, integrating center, and an effector. So all we're going to do is just talk about what the sensors are, what the integrating centers are, and what the effectors are. And I'm going to argue that you kind of already know all three of them for blood volume, and I'm going to talk about the ones for osmolarity. So let's look at the sensors for osmolarity. These are just called osmoreceptors. Osmo for osmolarity, receptors just for like sensors. So they sense osmolarity. And here's how they work. They're pretty freaking awesome. So for example, let's say that your blood osmolarity gets very high. As a result of that, water inside these cells, these special osmoreceptors, is going to diffuse out towards the higher osmolarity. Once that happens, the cells are going to shrink and basically shrivel up. When they do, the proteins on the cell surface are going to get changed in terms of their shape. If that protein is linked with a sodium channel, what can happen is then this sodium channel will open, allowing sodium to come into this osmoreceptor, triggering an action potential. Let's try that one more time. High osmolarity in the blood. This is the osmoreceptor, the inside of the cell. Osmolarity is high out here, water is going to leave, and the cell is going to shrink as a result of that water loss, which is what we already know should happen. The cell shrinks. This starred protein is going to change shape as the cell deforms. If this protein is physically linked with this sodium channel, it can then change the shape of this sodium channel, causing it to open. Sodium from the high blood osmolarity comes in from high to low. That sodium coming in then triggers an action potential, and now this osmoreceptor is super active, firing like crazy. And so when they're firing like crazy, these osmoreceptors send an action potential to the hypothalamus to stimulate the production or the release of ADH. Think to yourself real quick why we would want to release ADH if blood osmolarity was too high. I would wait. Take 5 seconds real quick. Or pause me. If blood osmolarity is too high, that must mean that we need to do either one of two things. We either need to get rid of some of that solute, or we need to dilute that solute with some water. ADH will allow us to do the latter. It will allow us to reabsorb more water to dilute this osmolarity. And so this is just a review of how these osmoreceptors are working. Increased solute or osmolarity outside of this osmoreceptor leads to them losing water. As a result of that, the cell shrinks. You can see it shrinking, getting smaller here. That triggers the opening of sodium channels, causing an increase in firing rate in this osmoreceptor. So that's how we sense a problem with osmolarity. Now let's look at how we sense changes in blood volume. Except you already know how. They're the same way that we sense changes in blood pressure. We're just going to use those baroreceptors just like we talked about last time, or a few weeks ago. Just to recap, these baroreceptors will be activated if the pressure in the blood vessel gets too high as a result of the volume getting too high. The neuron will stretch. That will physically open these sodium channels, and these baroreceptors will then fire an action potential. So for example, these channels will increase their activity if blood pressure and blood volume increase. These channels on the baroreceptors will decrease their activity if blood pressure and blood volume decrease. And just like the osmoreceptors, these baroreceptors will tell the hypothalamus whether or not to release ADH. All right, so now we have our first two tools to sense changes in these two very important parameters, osmolarity and blood volume. Now we're going to take these tools and apply them to see how we can affect the kidneys and all these other systems to regulate these two parameters. All right, so here's why I think this is so cool, because what we're about to do in the next slides is build this crazy ass complicated pathway that is going on inside of you right now, and it's going to look like a lot, it's going to seem really complicated, but you're totally going to know how this all works in the next 5 minutes, and much of it you already know. So for example, here's what we have so far. So we have some person that is super dehydrated. As a result, their blood volume and their blood pressure is very low. We also have the problem that their osmolarity is really high. So what we just learned is that blood volume and blood pressure will be sensed by baroreceptors, and we've seen that when we talked about blood pressure regulation in the past. But now what we're going to do is tell these baroreceptors to tell the hypothalamus to increase the production and release of ADH. ADH is going to tell the collecting duct to reabsorb more water. Blood volume is going to rise, blood pressure increases. So we have low blood volume, more ADH, increased blood volume. Problem solved. If we look at the problem with osmolarity, as we reabsorb more ADH, as we reabsorb more water as a result of ADH, that will also help us decrease osmolarity. So ADH is super powerful. It basically just cured two problems for us at once. Two birds with one stone. But just to make sure we fixed it, turns out there's another way that we can cause osmolarity to decrease. If osmolarity gets too high, the osmoreceptors can also sense that and tell the hypothalamus to also increase ADH to reabsorb more water. But what these osmoreceptors can also do, and what the hypothalamus can also do, is literally change your mood. It is going to make you feel thirsty and make you go get water to decrease osmolarity. So for some reason, for me, that's super trippy, that when we get thirsty, it's because these osmoreceptors are firing, telling the hypothalamus to tell you that you should feel thirsty and you should get your ass up and go drink some water because osmolarity is too high. We need to bring it down. So next time you feel thirsty, think about these ones. They're the ones that are helping you feel thirsty. Think about the hypothalamus. That is what is making you feel thirsty. It's crazy. All right, so we have this pathway, and I'm going to call this what's called the hypothalamic mechanism, because essentially we're affecting osmolarity and blood pressure and volume using the hypothalamus. But what we know, and what you already know, is that we can affect blood pressure in many other ways. We saw that the baroreceptors, in addition to stimulating the hypothalamus, like we just learned, can also affect the cardiovascular system. So we're going to add that piece now to this growing puzzle. Decreased blood volume and blood pressure sensed by baroreceptors, just like always. It will tell the cardiovascular control center in the medulla to affect autonomic nervous system output. So if blood pressure is too low, we want to increase sympathetic output, decrease parasympathetic output. By increasing sympathetic output, we tell the SA node to fire more, increasing the rate. We tell the ventricles to contract more strongly, increasing the force. By increasing sympathetic output, we tell the arterioles to constrict. And all these three will help increase blood pressure. These two by increasing cardiac output, this one by increasing blood pressure directly, by increasing the resistance to blood flow. So this one is just reviewed. That one we already know from back in the day. But just keep in mind that all of this is going on in the person that is hiking through the desert. Simultaneously, what they should be feeling, they should feel their heart racing. They should feel their ventricles contracting strongly. They should see that their blood vessels are constricting. At the same time, they should feel extremely thirsty. At the same time, they should be producing a crazy amount of ADH. And so basically your body is saying, it is go time. Like you are in a bad, bad situation. Blood pressure is too low. We're going to do everything we can to increase it. But you see how there's this big empty space here? That means that we're going to add another piece. I know, painful, but it just goes to show how important blood pressure and osmolarity is to your body. What we're going to do is to have the cardiovascular system control blood pressure, the hypothalamic system control blood pressure, and just to be safe, we're going to have a third fail-safe system just in case either of these fails. So what we're going to include now is the idea that the kidneys can also help directly sense and regulate blood volume and osmolarity. And here's how they do it. One way they can help regulate blood volume, as we saw last week, is if there's more filtration occurring here that means that more fluid will be lost into the filtrate, which means we'll pee more. So that's one simple way that the kidneys can affect blood volume. If blood volume gets too high, we can increase filtration here to pee out more stuff. So what we can do is increase or decrease what's called glomerular filtration rate to affect fluid loss. So for example, if blood pressure is too high, maybe because blood volume is too high, that will cause increased glomerular filtration, which means more fluid loss, which means that blood pressure will then start falling. So that's one way the kidneys can affect blood pressure and blood volume directly. Filter more out at the glomerulus. But another way is a little bit more complicated, unfortunately, but it is super, super powerful. Potentially the most powerful regulatory mechanism in your entire body. So let's see how this works. So here's the setup in this picture here. So you can see as they zoomed in onto the afferent arteriole, so basically zooming into this structure right here. The blood is coming in through here into the glomerulus and then leaving out through the efferent arteriole. And this tube coming up here is the ascending limb of the loop of Henle. So this right here is just a part of the nephron. And what you can see is that there are all these specialized cell types that they're pointing out. Here, these are called the macula densa cells. They're the cells that line